Okay, so some of this is going to be a review from what we already talked about in class, so you don't need to write it down again. Like homologous chromosome, we already wrote that down in class. I just want to make sure you remember, okay, these two chromosomes right here, they are homologous to each other. Why? Because they contain the same genes. And all the rest, every pair of chromosomes is homologous to each other. They have the same genes. You got one from your mom and one from your dad. Okay, so we're talking about sexual versus asexual reproduction. Remember, the bacteria and some other living things do cloning or asexual reproduction, where they just make exact copies of themselves, which is fast and easy, okay, but it gives no diversity. Okay, so sexual reproduction is much more advantageous because it gives so many different unique combinations of genes. Okay, this is where we get all the variety in life. Okay, if you look at these people here, all right, even the sisters, they look really alike, but there is small variations between them, whereas the brothers, maybe those don't look quite as similar, but yet they're brothers. Okay, same parents, unique variety of genes, and that's what meiosis is all about. Okay, again, don't need to write this down. You already have this in your notes. Okay, but when you have two copies of every chromosome, that's called a diploid cell. Gametes, which are your sex cells, the egg and the sperm, only have half the number of chromosomes, and so they're called haploid cells. Okay, meiosis makes haploid cells. So if you look at the picture here for just a minute, okay, meiosis occurs in the ovary and the testes to make the egg or the ovum or the ova, and in the testes to make the sperm. Okay, those gametes are now haploid. They have half the number of chromosomes, so that when they fertilize, we're back to a diploid number. Okay, so again, haploid, two haploids make a diploid. So again, the whole purpose of meiosis is to reduce the chromosome number from a diploid cell to a haploid cell. So how does it work? Okay, well, the chromosomes replicate, DNA replicates just like it did for mitosis, okay, but instead of the chromosomes lining up in a straight line like they did in mitosis, they're going to line up here with their homologous chromosome. Okay, so the chromosomes actually find their homologous chromosome, pair up with them, so that then they can actually get separated from each other. So that your homologous chromosomes with let's say, your gene for your hair color, okay, the chromosome you got from your dad's going to go over to one cell, the chromosome you got from your mom's going to go down into the other cell. So that you're only passing on your chromosomes that have one of every copy of every gene onto your kids. You can't pass both of your hair color genes onto your kids. You can only pass one of them on. And then a second division happens where the sister chromatids get separated and make the 4 cells, which are now haploid. Okay, so again, compare them with what we started with. Look at the first cell. It has 2 chromosomes. It's a diploid cell, and now we end up with 4 haploid cells. Okay. How does meiosis create so many unique variations? Well, one of it is the way that the homologous chromosomes pair up. Okay, look at possibility 1. Okay, the blue homologous chromosome lines up with the red homologous chromosome of the large pair, and then the blue and the red of the small pair. Okay, but when they separate, okay, we get the 2 blues over here and the 2 reds over here, and they separate again, and we end up with 4 haploid gametes. But there is another possible combination. So we could have the big blue one with the red small one and the big red one with the small blue one. Okay, and that's just a different possible combination. So you see in the end the gametes end up different. Okay, we get different combinations. So if the egg fertilizes the sperm, the other way around, the sperm fertilizes the egg, we get different combinations of genes. Okay, now this is only showing 4 chromosomes, so 2 pairs. Imagine in your cells you have 46 chromosomes or 23 pairs that are lining up like this. If you like math and you want to get some extra credit, you could figure out what is the possible number of combinations with 23 pairs of chromosomes. Okay, I'm going to show you some pictures that show some more detailed steps of meiosis. Okay, you do not need to write these steps down unless I specifically say to. So here we go. S phase, chromosomes duplicate. Replication occurs just like it did for mitosis. Okay, but now we're going to be in prophase 1 of meiosis. Okay, and something different happens here. This is where the homologous chromosomes pair up with each other. Okay, and this is what you do need to write down. So in prophase 1, crossing over occurs. This is when the homologous chromosomes pair up and exchange segments with each other. So if you look right here, okay, just like we saw in the video today, the homologous chromosomes have paired up and they're exchanging pieces of their chromosomes. That's called crossing over. And that's what creates even more diversity in the gametes, okay? So many different possible combinations, so many different ways to cross over that it creates way more variety. Okay, so now the cells are going to start to split their chromosomes, but it's going to be different. Now they're lined up with their pair, so they're lined up with their homologous pair during metaphase. They line up in the middle, okay? But notice how this is different, okay? They're getting separated from their homologous chromosome. That's called anaphase. So this is metaphase and anaphase, but it's different in meiosis. Okay, telophase is sort of the same. We start to reform the nuclei, okay? But now we have 2 haploid cells, okay, because they only have 1 of each chromosome. So a second division has to happen, which is called meiosis 2, okay, where basically the phases happen all over again. So prophase, metaphase, anaphase, telophase 1, and then prophase, metaphase 2, anaphase 2, okay, where we're lining up the chromosomes, and then the chromatids are getting pulled apart this time. And then we end up with 4 cells, 4 nuclei that are haploid, that are completely unique. Okay, so something to write down. Meiosis. Okay, you already wrote the first 2 bullets down, I believe, in class. So meiosis produces 4 cells that are genetically different from the parent cell and from each other. Okay, so not only are they different from the parent, but they can be unique. Even within every sperm is different, or egg. Okay, so again, just to compare the differences, in meiosis, the chromosomes find their pair, and they line up in pairs, whereas in mitosis, in metaphase, they line up straight up and down in the middle, and they're just separating from their sister chromatid, so they can separate the replicated DNA. Okay. Here they're separating from their homologous chromosome so that they only pass on 1 copy of every gene to their offspring, and then the sister chromatids split again, and you end up with 4 haploid cells. Okay. So meiosis is so great for creating variations, okay, and so independent assortment is how the chromosomes can line up anyway, crossing over when the pieces exchange, and then random fertilization just means that any sperm can fertilize any egg, and you can get different combinations every time. Okay, so last review. Homologous chromosomes pair up. They might cross over. The homologous chromosomes separate from each other, and then the sister chromatids separate, and you end up with 4 gametes, again sex cells, that are haploid. They have half the number of chromosomes, and they're unique. Notice how none of these 4 cells is the same. They don't have the same DNA anymore. This is what's going to create the unique variations in our offspring, and that's it.